This conference will now be recorded.
Also, I wish you all a happy Dasara. So, in the last classes, what all things we were learning already, some of you might know, but I think today we have got few new members. Maunia, Milisha, yeah, Rita Brota Basak is a new member. Shalini, then Shanmati is again new member, and I think Smita Kumari also new member. So before going to the topic, let me know whether you have gone through the previous classes in YouTube, any of you, because when I started with microscopy in the first class, I took basic terms in microscopy. Then in the second class, we started with light microscopy, and we have completed bright field microscope, dark field microscope, and fluorescent microscope. So today we are going to electron microscope. Before that, we have one more topic, phase contrast microscope. So what I expect is those who have attended the classes, they can very comfortably continue this lecture. Whereas those who are new to this topic microscopy, they will find a little difficulty in catching up with the points. So what you can do is either after this class, please visit the YouTube channel, or even they have shared the link in the Telegram as well. Just hear the previous two classes on microscopy and then come back and learn these topics so that it will be easy for you. But anyway, I will try my level best to make you all understand regarding today's topic. And if you have any issues, any suggestions, or you want me to explain more or anything like that, please let me know in the comment box. Fine. Thank you. Yes, so today we are going to start with the fourth type of light microscope. So just a review of the last few classes. We started with the terminology, fine, basics, then about some terms in microscopy, all those things we have covered actually. Fine, about the different parts of a microscope, then various terminologies again. Then we started with light microscope. We completed bright field microscope, dark field microscope, then fluorescent microscope. So today we are going to take the fourth topic, that is that's fine, Millie. That's fine. You please go through the YouTube channel. That is more than enough. Okay, that's fine. That's fine. Because it is up to you. Because this class is as per your convenience. So those who can attend live, please attend live. Otherwise, you can even go through the YouTube channel and continue if you have any emergency. That's fine. Okay. So today we are going to deal with the fourth type of light microscope. Before going to electron microscope, we will complete the light microscopy, and then we will go to electron microscopy. Fine. So we completed bright field microscope, dark field microscope, and phase contrast and fluorescent microscope. You all remember all those things. In bright field microscope, the field appears bright, whereas the organisms appear dark or stained. Whereas in dark field microscope, the field appears dark, but the organisms appears bright. And in fluorescent microscope, of course, the organism will appear in fluorescence due to staining with fluorescent dyes. All those things we have covered. So this is the another type of light microscope. Now, why I took this topic is, in your syllabus they have clearly mentioned microscope, visualization of live microorganism. So while we were taking this topic, before going to phase contrast microscope, while we were explaining about bright field microscope, we told this is mainly used for stained specimens. You can view here. See, bright field microscope is mainly used to view stained specimens, whereas dark field microscope is used to view unstained specimens, eliminate staining. Unstained specimens can be viewed using dark field microscopy. And in fluorescent microscope, we have got fluorescent dyes, by which if we are illuminating, if we illuminate the specimen with blue light, it will emit green light, and even it can be conjugated with antibodies. All those things we have discussed. Fine. So now, what is the specialty of our today's topic, that is phase contrast microscope? Staining is not required to view phase contrast microscope. Staining, so that is the first thing you have to understand about phase contrast. Staining is not required. We don't need staining, which means it can be used to visualize live microorganisms. So that is the most important point of phase contrast microscope. We can visualize live microorganism using phase contrast microscopy. Even in bright field microscopy also sometimes we visualize live microorganism. Hope you all remember about motility testing. Have you done motility testing in your college? Can I get the answer in comment box, yes or no? Motility testing, observation of microbial motility or bacterial motility. How many of you have done in the college? Stab agar, not even stab agar, using microscope. Using microscope, have you done motility? Using bright field microscope, we can observe motility. There is an experiment for that. We can see even E. coli, motile organisms like E. coli, or even Pseudomonas, or any motile organisms we can visualize. Okay, you might have not done. Okay, just leave it. I just wanted to ask you whether you have done it. Fine. So here in this light microscope, in this phase contrast microscope is a sophisticated type of light microscope which is mainly used to visualize live microorganism. So this is an important thing because in the syllabus itself they have given about this topic. You just go through the syllabus. In unit 13, I think part F or G is microscopy. There they have given this point about live or about visualization of live microorganism. So that's why I selected this microscope as well. Fine. So phase contrast microscope, the first thing what we want to know, staining is not required, as well as it is used to observe live microorganism. Now, how phase contrast microscope works? Okay. Now, light that passes through a cell or structure is slowed down relative to light that passes directly through the less dense surrounding media due to difference in diffraction or even refraction, we can tell. Fine. So just imagine we have a slide on which we have the microbial culture. Light will be passing through the slide. Light will pass through the edges of the glass slide with a speed, but when the light hits the middle portion, that is the portion where we have microorganisms, it will slow down comparatively. That is, light which passes through the cell or structure is slowed down when we are comparing with the light that passes through the less dense surrounding medium. It is due to difference in either diffraction or even refraction. So then comes the two questions: what is diffraction? What is refraction? That's why I took all these terms in the first classes. Can anybody answer in the comment box? What is diffraction? What is refraction? What is refraction? What is diffraction? Anybody can answer. Can anybody answer in the comment box? What is diffraction? What is refraction? Anybody? Anybody? Those who have attended my first class, or anybody those who have gone through the YouTube channel, you can very well answer. So just make these terms clear for you. Now, refraction is bending of light. Okay. So Jancy and Shalini, thank you for that. Refraction is nothing but the bending of light when it passes from one medium to another. For example, when light enters from air to the glass slide, it will lead to bending. It may cause bending. It may result in bending of light, which is called refraction. And then we came to the term refractive index as well. Fine. So whereas diffraction is splitting up of a light into different wavelength, which is nothing but diffraction. And diffraction may even lead to blurring of image. Clear? All those things we have discussed in the first class. Fine. Thank you. So here what is happening in phase contrast microscope, the light which passes through the cell or structure or through the specimen will be slowed down when comparing with the light which passes through the surrounding glass slide or edge of the glass slide surrounding less dense medium. Okay, now what is happening? Now this refracted or diffracted light, it can be either refraction or diffraction. I have written only diffraction, but it could be either diffraction or refraction. Okay, now the diffracted or refracted light by the specimen when passes through a region of phase plate that alters its phase. So there is a structure in there was a special structure in dark field microscope. Due to that structure, we had only light which is reflected by the specimen will only enter the objective lens in dark field. While we study dark field microscopy, I told you about a structure or a fitting or a special aid on dark field microscope. So the light which is reflected by the specimen will only enter the objective lens. Can anybody tell me what was that structure on dark field microscope? There was a special structure on dark field microscope which will not permit the direct entry of light through the condenser lens. And in dark field microscopy, there is another speciality. The light which is reflected by the specimen will only enter the objective lens. There was a special structure. Very good, Shalini. Dark field ring. I am not sure whether you are googling it or whether you are going to my previous slide. I don't think so. I trust you. So very good, dark field ring. Very good, Milisha, Shalini. So in dark field microscopy, we had a structure, special structure called dark field ring, which was not permitting the entry of light into directly through the condenser lens. So here in phase contrast microscope also we have got a structure called phase plate. That is a special structure in phase contrast microscope. It is called phase plate. So what is the function of phase plate? Now, first of all, the light will be refracted or diffracted by the specimen. Okay. As a result, the speed will be slowed down. Fine. Okay. Understand. Now, this refracted or diffracted light by the specimen will pass through a region of phase plate. It will pass through a special region called phase plate region. Now, what is the function of phase plate? It will alter its phase. Again, it will change its speed or alter its phase. Fine. That means the greater the refractive index of the cell or structure, the greater the retardation of light wave. So again comes the term refractive index. What is refractive index? Anybody in the comment box, what is refractive index? Refraction is bending of light when it enters from one media to another. For example, from air to glass slide. That is a refraction. Now tell me, what is refractive index? What is refractive index? Anybody in the comment box? Anybody can type. Yeah, ratio of speed of light from given medium to vacuum. Very good. Ratio of velocity of light. Very good. So refractive index is nothing but when we compare the ratio of speed of light with a medium. For example, if we pass light through the glass and we measure the speed of the light which is passing through the glass, the same intensity of light when we are passing through vacuum. Vacuum means absence of air or absence of oxygen. We will be getting a value. The difference between these two values is nothing but the refractive index. So again, we are coming back to phase contrast microscope. The greater the refractive index of the cell or structure, the greater the retardation of light wave, which means if the refractive index of the microbial cell or the structure which we want to observe is higher, the retardation of light wave or the speed of light wave will be retarded higher. But if the refractive index of cell or structure is lower, the speed of light will be also comparative to that. So based on the refractive index of the cell or structure, the light wave will be retarded or the speed of the light wave will be altered or the phase of the light will be altered. Fine. Now finally, the differences in phase altered into differences in intensity result in a visible difference in contrast between the specimen and the background. So this is finally happening in the phase contrast microscope. The differences in phase. So already we know the light which is passing through the microbial cell is slowed down when we are comparing with the light which is passing through the less dense surrounding media or the glass slide. Now this results in refraction or diffraction will be converted into a phase change which alters its phase, which alters its speed by a special structure which is present in phase contrast microscope called phase plate. Fine. Now this differences in phase is altered into differences in intensity, which means if the speed of light is reduced, definitely its intensity will be reduced. If the speed of light is higher, intensity will be higher. So all these things are correlated, related with one and another. So here finally what is happening? The differences in phase is altered into differences in intensity, which results in a visible difference between the contrast, which results in a visible difference in contrast between the specimen and the background. So as a result of this, we can observe the microorganism in a background. Clear? So this is the phenomenon of phase contrast microscope. It is very simple. If we are explaining it in a very simple method, I can tell you. On the glass slide, you will be having a microbial cell or microbial smear or whatever it is. Light will hit on the slide. Light will pass through the microbial cell also. Light will pass through the unstained region or the side of the glass slide also. The light which is passing through the microbial cell will be slowed down more than the light which is passing through the edge of the glass slide or the unstained region. Now, this due to the difference in refraction or diffraction, because glass slide, when light is passing through the glass slide, it will be refracted or diffracted in a different way when we are comparing with the light which is passing through the microbial cell. Now, this light, this diffracted or refracted light by the specimen is then passed through a special area called faceplate region or a region of faceplate or faceplate, which alter its phase, which again alter its intensity or phase or speed. Now, if the refractive index of the cell or structure is higher, the amount of retardation will be also higher. If it is lower, the amount of retardation will be lower. Based on the refractive index of the cell or structure, the light wave will be retarded or the intensity of the light wave will be reduced. Now, this difference in phase or this difference in speed of the light wave is converted or altered into differences in intensity, which results in a visible difference in contrast between the specimen and the background. Contrast, you know what is contrast? Fine. Can anybody answer what is contrast in the comment box? Anybody? What is contrast? Contrast. Already we have learned this topic. Contrast. What is contrast? How? What we do in order to increase the contrast? What is the technique we perform in order to increase the contrast? Difference between specimen and the background is contrast. Yes, Charlie, very good. Now tell me, what is the technique? Very good. Staining. Why we are performing staining? If we are not performing staining, the microorganisms which is colorless will appear in a colorless background and you cannot identify which is the microbial cell, anything. So in order to increase this contrast, in order to differentiate the microbial cell, we are performing staining. So staining is a technique by which we can improve the contrast. Fine. So here, finally, what is happening? The differences in phase is converted into differences in intensity, which results in a visible difference between the difference in contrast between the specimen and the background. Fine. That's about phase contrast microscope. So this is the way we can see the light source which is entering through the condenser lens, unobstructed light. See, phase unaltered by specimen. Here the light is entering through the light source which is entering through the condenser lens. When it hits the specimen, here this is the specimen, it hits the specimen. Now refracted or diffracted light, that is phase altered by specimen, this light will be refracted or diffracted. But now the refracted or diffracted light by the specimen will pass through this region which is face plate. See, here you can see the face plate. So the face plate will alter this difference into difference in phase or difference in speed. Now, depending upon the refractive index of this cells or the specimen, it will produce a difference in intensity, which will give you a proper contrast between the microbial cell and the surrounding media. Clear? This is what happening in phase contrast microscopy, and like this microorganisms. So here in bright field we told microorganisms will appear bright in a dark background. In dark field microorganisms will appear dark in a bright background. In fluorescent it will appear fluorescent. So this is the way. So it is almost a microorganisms will appear as live structures in a colorless background. So you can see from the picture how microorganisms are appearing, how the cells are there, how the organisms are appearing, and all those things. Clear? So that's all about phase contrast microscope. Now, before going to electron microscope, let me ask you if you have any doubt from the phase contrast microscope. Is it clear? Phase contrast microscope, what is happening in phase contrast microscope? Simple actually. The terms are complicated, but once you know the basic terms of a microscopy and all, it is simple, because I have made the topic as simple as possible. If you refer many textbooks and all, you can find a lot of things, but this is the simplest version how you can understand. That's what I have made it as much as simple as possible. More than this is not possible, because anyway we can expect some multiple choice questions, so we must know, because every microscope has got its own speciality. So based on that, they will be asking. And I hope you have gone through all the questions which I have asked in the Telegram group. Just try to answer those questions so you can get some answers and some explanations in the bottom. So that will also help you. Fine. So with I hope so I got answer from two of you, Jancy and Milisha. It is okay for both of them. Now, what about the rest? Regarding Rita, Brota, Baset, because you are a new participant, then Shalini, Shanmati, Smita Kumari, Khan Shaista, Kanimuri. Kanimuri is also a new participant, it seems. Maunia, she was there in my first class. I am not sure about second class. Then Priyama Kumari, Milisha, all of you were there, some of them. So I hope it is clear for most of you. If you have any doubt, please do let me know. Fine. I have already shared my WhatsApp number and email ID, everything. Fine. Clear. So with this our topic light microscope comes to an end. Now we are moving on to our today's topic. We have to finish this microscopy by today. Fine. So that one portion from your unit 13 will be completed by today, and we can move on to another portion by next class. So now we are going to the second type of microscope, electron microscope. So in the beginning itself, I told you two types are there, basically light microscope and electron microscope. In light microscope, we are using light source to illuminate the object, whereas in electron microscope, we are using a beam of electrons to illuminate the object. Fine. And there are a lot of other differences based on magnification. When it comes to magnification, light microscope can provide you a maximum magnification of 1000x, that is 1000 times. And if we are going for oil immersion objective, we may get a magnification of around 1400x or 1400 times, whereas in electron microscope the magnification is quite higher. It's around 10 lakh times, more than 10 lakh times the magnification. Yes, that's fine, Priyama Kumari, you are a bit late. That's fine, that's fine. I hope you are following. And then about the resolution of a light microscope, it was around 200 nanometers, whereas in electron microscope it will be quite higher. So now we are moving on to electron microscope, right? So those who know the basics of light microscope and microscopy, electron microscope will be very easy for you. But those who don't have any idea about the terms of microscope and microscopy as well, it will be a bit difficult, but I will try my level best to make it simple as possible. Fine. Now electron microscope. So what is an electron microscope? First of all, a beam of electrons instead of light. So here, instead of light in light microscopy, we are using a beam of electrons to illuminate the object. Now, greater resolution. So we are getting a very higher resolution in electron microscopy when we compare with light microscope. Because why? Wavelength is much shorter than light. Shorter wavelength, you can see. So in light microscopy itself, while I was explaining, I told you, as the wavelength of the light decreases, the resolution or revolving power increases. Resolution or resolving power increases. As a result, you are getting a better view of the specimen. Similarly, in electron microscopy, so for example, in light microscopy, blue light, which is having a wavelength of around 480 nanometer, will provide you a better resolution or resolving power than a red light, which is having a wavelength of around 680 nanometer. So all those things we have explained in light microscope. So when it comes to electron microscope, the wavelength of electrons are much shorter than the light, which means the resolution will be quite higher than the light microscope. Fine. As a result, you are getting a higher magnification as well. That is, at 60,000 volt, the wavelength is approximately 0.005 nanometer. So wavelength of electron beam will be around 0.005 nanometer at 60,000 volt with a resolution of 0.2 nanometer. That is 1000 times better than the light microscope. So in light microscope, for example, when we are taking blue light, we were having a wavelength of around 480 nanometer, which was providing us a resolution of around 200 nanometer. That is, most of the light microscope, the magnification is around 1000x and resolution 200 nanometer. So when we are using an oil immersion objective, it will go up to 1400x. Whereas in electron microscope at 60,000 volt, because high voltage or high volt of electricity is required to produce an electron beam. When we learn about the electron microscope, we will learn more. Fine. When we are continuing, you will learn more about that. So in electron microscope, the wavelength of electrons are at 60,000 volt is approximately 0.005 nanometer. As a result, with a higher resolution of 0.2 nanometer, that is 1000 times better resolution than the light microscope. So when we are using electron microscope, our resolution will be 1000 times better. Fine. So in light microscope, it is around 200 nanometer resolution, whereas in electron microscope, it is around 0.2 nanometer resolution. In light microscope, for example, if we are using blue light, the wavelength is around 480 nanometer, whereas in electron microscope, if we are using beam of electrons, producing a beam of electrons at 60,000 volt, the wavelength will be approximately 0.005 nanometer. Clear? So the resolution of electron microscope is 1000 times better than that of the light microscope. Clear? Now, as a result, the magnification is quite higher than the light microscope. In light microscope, the magnification was around 1000x or 1000 times, whereas here it is 1, 10, 100, 1000, 10,000. 1, 10, 100,000, 10,000 lakh. Okay, there is a mistake here. It is not 1 lakh x, it is around 10 lakh x, more than 10 lakh x or 10 lakh times. In the first slide itself, I have explained this point. So here I think 1 0 is missing. 1, 10, 100,000, 10,000 lakh. No, not 1 lakh x, it is around 10 lakh x. In the first slide, I have explained this thing. See, 1, 10, 100,000, 10,000 lakh, 10 lakh x. Electron microscope, the resolution is up to 10 lakh x, 10 lakh times. Fine. Whereas in light microscope, the resolution is up to 1000 times. Light microscope is used to observe bacteria, algae, fungi, protozoa, that is basically the whole cells. Whereas electron microscope is used to observe viruses, bacterial cell structures, etc. Fine. This was the first thing we explained in the class. Clear? So coming back to our electron microscope. Yes. So it is. So that's all about the comparison of light microscope and electron microscope. In light microscope, the wavelength will be, if you are using blue light, the wavelength will be approximately 480 nanometer, whereas in electron microscope at 60,000 volt, we can produce a beam of electrons with a wavelength of approximately 0.005 nanometer. Now, in light microscope, the resolution, maximum resolution possibly is around 200 nanometer, whereas in electron microscope, having a resolution of around 0.2 nanometer, that is 1000 times better than the light microscope. Fine. Whereas in case of magnification, in light microscope, the magnification is up to 1000x, or when we are using oil immersion objective, the magnification will go up to 1400x, whereas in electron microscope, we can get a magnification of around 10 lakh x or 10 lakh times or even better magnification using sophisticated electron microscopes. Even it can go up to 20 lakh x. Fine. Now, the most important thing in electron microscope we have to learn is about the specimen preparation. It is also mentioned in your syllabus regarding specimen preparation and staining used in electron microscope. We have to learn a bit about that. Whereas in light microscopy, you will be learning it in another portion regarding staining and symbol staining, gram staining, all those things you will be having. I am not sure about that. Fine. So in electron microscope, now we are passing on to the first stage, that is specimen preparation. How we are ready with a specimen for electron microscopy. Now, the improper sample formation or improper sample preparation will lead to artifact formation. So, if we are not preparing our specimens properly in electron microscopy, it may lead to a phenomenon called artifact formation. What is artifact formation? Appearance of something in an image that is not a true representation of specimen features on view. Okay, that is artifact. That means, for example, in light microscopy as well, when we are performing some staining, sometimes we can observe in light microscopy, we can observe stained areas, but it will not be the microbial cell. It will be due to the improper staining or something. It may appear as a microbial cell. Similarly, in electron microscope, if we are not preparing the sample or if we are not making the specimen properly, it may lead to a phenomenon called artifact formation, which is nothing but appearance of something in an image that is not a true representation of specimen features on view. While viewing, we will be misguided that it is the microbial cell structure, but it won't be the microbial cell structure. It may be an improperly prepared specimen. That is, it will imitate as a cell structure. The specimen will imitate like a real specimen or real cell, but it won't be that. It will be due to the problems in our sample preparation. Fine. So that is called artifact formation. Now, what we have to do? So before view, what we are doing? We are dehydrating the specimen. It is necessary to dehydrate the specimen. Dehydrate means remove the water. To fix, that is preserve intact the structures. So we have to fix the specimen. In while light microscopy also we are doing what all things we are doing in light microscopy. Sometimes we are doing air drying. Sometimes we are doing heat fixing while we are performing gram staining. So here also in electron microscopy also some of these steps will come in specimen preparation. For example, first of all, dehydration of the specimen, that is removal of water from the specimen. Then fixation, that is to preserve the structures intact. We have to make sure the microbial structures are maintained intact in position in the specimen. For that we are going for fixation, that is by natural orientation. So why we are doing fixation? We make sure the microbial cell structures are preserved or are present in position intact in the natural orientation. For example, if the microorganism will have flagella, if it is a diflagellate organism, it is having two flagella. Okay, the flagella will remain intact in the position in front of the cell. It will not be removed during fixation. So all those things we have to. Yes, I have got a doubt in the comment box. Okay, what is the difference between heat fixing and dehydration? Dear, dehydration is just the removal of water from the cell. If we are removing water from the microbial cell, it is not necessary we have to do dehydration by heat fixation. In heat fixation, we make sure first of all the microbial cells will die definitely, and we fix it on the slide so that during our staining or subsequent washing step, these cells will not be removed. So in heat fixation, we make sure that the cell structures which is fixed on the slide will not be removed during staining or subsequent washing step. Whereas if we are dehydrating the cell by maybe simple air drying or something, and we are going for staining and washing, there is a chance microbial cells will be removed. Clear? In electron microscopy, we are going for dehydration. There is a special technique for that. I will explain in the next slide. What is the way by which we are going for dehydration? I will explain in the next slide. Fine. So here we are dehydrating the specimen, then fixing the specimen in order to make sure all the microbial structures are present intact. Then now see, during fixation, it is possible to shrink or stretch or distort. So all those things we have to make sure it is not happening. Shrinking of the specimen can happen, then stretching of the specimen can happen, distortion of the specimen can happen. So all these steps must be removed, or all these problems must be taken care of when we are doing fixation. Okay, now if this is happening, if shrinking is happening, or if stretching is happening, or if a distortion of image is happening, what is happening? The image will be altered, the image will be changed. So when we are going for specimen preparation, especially in electron microscope, we have to take utmost care. We have to dehydrate it, we have to fix it intact in order to make sure that the microbial structures are present in the natural orientation. We will not lose any microbial structures in the original specimen. Fine. So also we have to take care whether the specimen will not shrink or stretch or distort, which may lead to the alteration of the image. Clear? So that means we have to carry out the specimen preparation in electron microscope with utmost care, even more and more careful than light microscope. Fine. Now these are the different methods by which we are going for specimen preparation in electron microscopy. It can be either thin sectioning and staining together, or it can be by means of freeze etching as well. Various techniques. First of all, in thin sectioning, what we are doing? If we are going for thin sectioning, first of all, the microbes are embedded in a plastic resin. So we are covering the microbial cells with a plastic resin. Microbes are embedded in a plastic resin prior to thin sectioning. So from the time itself, you know, thin sectioning means we are going to convert the microbial structures into very thin sections, very thin portions. So for that, before going for thin sectioning, we will cover the microbial cells, or we will embed. Embed means covering. We will completely cover the microbial cell structures with a plastic resin. You will be providing a plastic resin coating over the cell surface. Clear? After that, we are going for slicing. Sliced into thin sections before staining. So before staining, we will slice it into thin sections using a structure, or using a using an equipment called microtome. So what is microtome? Microtome consists of microtome is an equipment with a knife-like surface made up of diamond or glass. Okay. So first of all, we select the microorganisms which we are going to observe in electron microscope, whether they are bacterial cells or viral cells or whatever. We are covering it or embedding it in a plastic resin or plastic coating. Just understand, we are providing it a plastic coating. After covering with plastic, or after providing a plastic coating, we are cutting or slicing it into very thin sections before staining using an equipment called microtome. What is microtome? Microtome is an equipment which is used to produce the thin sections in an thin sections of microorganisms in an electron microscope. Okay, it is a knife-like surface made up of diamond or glass. Fine. And the resulting specimens will be having a size of around 600 to 700 nanometer in depth. Fine. So by this, now we are having thin sections of microorganisms with us. After this, we stain the sections with heavy metal containing components, such as, for example, phosphotungstic acid containing heavy metal tungsten. So why we are going for heavy metal components? Because we are not going to use light waves in order to illuminate the object. In electron microscopy, we are going to use a beam of electrons. As a result, staining has to be performed with heavy metal containing compounds. For example, such a compound or such a heavy metal containing compound which we use in electron microscopy, example is phosphor tungstic acid, which contains the heavy metal tungsten. Clear? So that's what thin sectioning, and now we are going to staining. So this before staining we are performing thin sectioning, and after thin sectioning we are performing staining using for example phosphor tungstic acid. Clear? Now staining is again used to improve contrast. Already I have repeated it several times, but whether it is gram staining or symbol staining or staining for electron microscopy, whatever, our purpose is to improve the contrast in order to distinguish the microbial cell structures from the background in a proper manner. Fine. Now here we are using stains contain electron dense heavy metal salt which scatters electron beam. So that is the purpose why we are using heavy metal containing compounds. We have to use stains which contain electron dense heavy metal salt. They are electron dense in nature. Fine. So that the electrons will be scattered, or an electron beam will be scattered at high voltage. Clear? And the stained areas appear dark or black. Usually in electron microscope, the stained areas will be appearing in dark or black. Clear? So thin sectioning and staining is a type of that is a method used for the sample preparation in electron microscope. Now instead of this thin sectioning, we have got another method called freeze etching. Freeze etching combined with staining can also be used. Now what is freeze etching? Here what we are doing. So in freeze etching, it is first of all used to define various biochemical layers of microorganisms. Why we are performing freeze etching? In order to define various biochemical layers. For example, when you come to your gram-positive bacteria, it is having a thicker peptidoglycan layer. Fine. When it comes to gram-negative, you are having a thinner peptidoglycan layer. Then in gram-negative bacteria, in addition to thinner peptidoglycan layer, you are having phospholipids. So these are the various biochemical layers present on microbial cells. So if we want to define various biochemical layers of microbial cells, including the organelles, including the internal organelles such as mitochondria, Golgi bodies, endoplasmic reticulum. For all those things, we have to go for freeze etching. So what is the primary purpose of freeze etching? In order to define or in order to distinguish between various biochemical layers of a microbial cell, and also in order to properly view the internal organelles. We are going for freeze etching. Here what we are doing, first of all, the specimen is first frozen at -196 degrees Celsius. First of all, the specimen or the microbial cell structure or the microbial cells which we are going to observe is undergo deep freeze at -196 degree Celsius. After that, it is fractured by striking with a knife blade. So similarly, similar to our thin sectioning, we are going for, after first of all, we are freezing the specimen up to -196 degrees Celsius. After that, we fracture the specimen, we section the specimen. Similar to the thin sectioning, we will convert the specimen into various sections or various portions by striking with a knife blade, and this process is called freeze fracture. It is a phenomenon included in freeze etching, freeze fracture, fracturing of specimen by striking with a knife blade. After that, itching. Itching means something like fixing. Itching is performed by raising a part of surface layer of the specimen. So we are going to, so which area, if we want to observe the cell wall, we are performing proper itching. We are improving, or we are raising the area of cell wall from the background. So itching or fixation is performed by raising a part of surface layer of the specimen. Okay. After that, then it is exposed to vapors of a heavy metal. That is, we are going for staining. First of all, we are freezing the specimen at 196 degrees Celsius. Then we are going for freeze fracture, that is fracturing the specimen by striking with a knife blade. After that, we are fixing or etching the specimen in order to raise the surface layer of the specimen. Fine. Finally, we are exposing it to vapors of a heavy metal such as phosphotungsten or phosphotungstic acid. Then after that then the specimen is rotated and exposed to vaporized carbon. After going for vapors of heavy metal, again we are exposing the specimen with vapors of carbon or vaporized carbon at 90-degree angle to produce a replica of the surface. So the purpose of freeze etching is to produce a replica of the specimen. We are producing a replica of the specimen using freezing, then fracturing, then etching, then staining by means of heavy metal vapors, then by exposing to vaporized carbon at 90-degree angle. Finally, we are producing a replica of the specimen, and any adhering biological material will be removed and the replica is observed. So here the original microbial cell structures will be finally removed. Once we get a replica of the specimen by all these methods, we will remove the biological material. That means the cells will be removed and this replica will be observed by means of electron microscope, using electron microscope. Clear? So that is about the specimen preparation of electron microscope. And finally, the last portion of the presentation, that is about two types we have electron microscope. We have two types of electron microscope. That is, first one is transmission electron microscope and second type is scanning electron microscope. Fine. So now we are going to, first of all, transmission electron microscope. Before going to transmission electron microscope, till here, if anybody is having any doubt, yes, sir, when striking with a knife cell will not get damaged. No, it's not like that, because even though we have written like it is fracturing by striking with a knife blade, means the purpose is to produce thin sections of the specimen. That is the purpose. Like how I have explained in thin sectioning, we are slicing it into thin section. Here also, even though we are striking it with, because it is written, it is explained like it is fractured. Fracturing means making it as portion, making it as similar like thin sectioning. Because in light microscopy, we have to observe the microbial cell as a whole. Here we are going to observe the internal organelles of the microbial cell, various biochemical layers of the microbial cell. In that case, the whole microbial cell, we must get some thin sections of the whole microbial cell. Then only it is possible to observe the biochemical layers and even the internal organelles. So whether thin sectioning using microtome or in freeze fracture, in both methods, the purpose is to produce thin sections. So here, if we, which means when we explain like it is fracturing by striking with a knife blade, the purpose is to produce thin section. That's it. We have to get thin sections. So that's why we are going for slicing into thin section. Here we are going for fracturing by striking with knife blade. The purpose is the same. I hope it's understood for you. The purpose of both these methods, whether we are using microtome or whether we are going for freeze fracture, our purpose is to produce thin sections of a specimen. Clear? Now, when striking with knife cell, will not get damaged. Maybe it may get damaged, because see, in thin sectioning already we have explained, the specimens are embedded in a plastic resin. So you will be having a plastic coating over the specimen in thin sectioning. If the plastic coating is not as thick as it is, it will be having a particular diameter, thickness, or particular size and all, molecular size and all. If it is not that much thick when we are going for thin sectioning, maybe we will get some artifact formation. That's why I explained in the beginning. What is artifact formation? Artifact formation is nothing but if we are not preparing the specimen as per the required parameter, there is a possibility that we will, when we are observing through the electron microscope, we will misunderstood. We will misunderstand a improperly prepared specimen with some microbial cells. Clear? That's why we have to make sure that in electron microscope the specimen preparation has to be done very precisely, because any of us don't have any experience doing electron microscope specimen preparation, right? It is not a common thing. Electron microscope is only present in that much research institutions, virology labs, or it will be not much, because it is like a highly sophisticated equipment, very costly. In such a those organizations which can maintain the cost only, they will buy an electron microscope. Clear? So that's all about the specimen preparation of electron microscope as well. So any more doubts till this area? Electron microscope introduction, then electron microscope specimen preparation. Is it clear for you all? Can you please type yes in the comment box if it is clear, so that I can pass on to my last portion. Is that clear till here? Okay, Jansi, Mili, Shalini, Shanmati, Darshana, Datta, thank you so much. With this, we are going to the types of, Maunia, we are going to the different types of electron microscope. So already I told you in the beginning, two types of electron microscopes are there: transmission electron microscope and scanning electron microscope. I will explain both of them, then you will understand the difference. I am not going to tell directly the difference. I will explain both of them. What is transmission electron microscope? What is scanning electron microscope? Now, how the image is produced in this both microscope, everything it is same. What is the phenomenon? Everything is same, but there is some difference. Fine. So we will start with transmission electron microscope. Now, what is transmission electron microscope? What we are doing here? First of all, it uses an electron beam and electron magnets instead of light source and glass lenses in microscopy. So whether it is a transmission electron microscope or a scanning electron microscope, both of them, any electron microscope, it uses an electron beam in order to illuminate the object instead of light source in light microscopy, and it uses electromagnets in order to direct the electron beam onto specimen instead of our lenses in light microscope. Fine. So electron microscopes, whether it is transmission electron microscope TEM or scanning electron microscope SEM, both of them uses an electron beam and electromagnets instead of light source and glass lenses in light microscopy. Clear? Now, what is the source of electron? The source of electron is a hot tungsten filament in an electron gun. So the source of electron in transmission electron microscope or even scanning electron microscope is a hot tungsten filament. It will be around, as in the previous slide I have explained, what is that, 60,000 volt, something like that. Very high voltage we are applying in electron microscopy. So here, that's why the tungsten filament will become hot. So the source of electron here is a hot tungsten filament in an electron gun. Now what is happening? Heat draws the electrons from the filament. So as a result of application of high voltage of around 60,000 volt, the tungsten filament will become hot, red hot, and electrons will be eliminated, or electrons will be drawn from the tungsten filament. Now these electrons are accelerated as a fine electron beam. So you are getting a fine beam of electrons as a result of this, and it is focused on the specimen with an electromagnetic condenser. So there, the normal condenser lens in light microscopy, the normal condenser lens will focus light on the specimen, whereas in electron microscope, the fine beam of electrons produced by the hot tungsten filament as a result of application of a high voltage of around 60,000 volt will be focused on the specimen with an electromagnetic condenser. Clear? Then the air is removed from the electron path by a high efficiency vacuum in order to avoid collision with gas molecules. So we are producing a fine beam of electron, and the fine beam of electron are directed onto the specimen by means of an electromagnetic condenser. Now, in the path of the electron, if there are air particles, there is a possibility that the electrons will be collided with the gas molecules, so that the path of the electrons will be altered, so that we won't get a fine image. In order to remove this issue, or in order to eliminate this issue, the air in the path of beam of electrons are removed using a high efficiency vacuum, so that no collision with gas molecules will take place. Clear? Now, as a result of this electron microscope, or when we are using transmission electron microscope or TEM, fine details even to the level of molecular arrangement can be observed using a transmission electron microscope. We will get a fine details of the specimen, that is, even to the molecular arrangement of specimen can be observed using transmission electron microscope. So that is about transmission electron microscope. Now, how the image is produced, I will explain in the next slide. Okay. So basically, in transmission electron microscope, the mechanism is very simple. Whether it is transmission electron microscope or scanning electron microscope, both of them uses an electron beam and electron magnets instead of light source and glass lenses. Now, the source of electron is a hot tungsten filament. How the tungsten filament is made hot? By applying a suitable voltage, maybe around 60,000 volt. As a result of the hot tungsten filament, as a result of tungsten filament becoming hot, the heat will produce a beam of electrons from the tungsten filament, now which is accelerated as a fine electron beam and focused on the specimen by means of electromagnetic condenser lens. Now, if there is air in the path of electron beam, collision with gas molecules will take place and the path of the electron beam will be altered. In order to overcome this, the air is removed by means of a high efficiency vacuum, and fine details even to the level of molecular arrangement can be observed using a transmission electron microscope. So transmission electron microscope means electrons are transmitted, which will be focused on the specimen by means of an electromagnetic condenser lens, and the image is produced. Whereas, okay, now this is about the electron path in a transmission electron microscope. We can see here. See, there is an electron source or electron gun. You have got an electromagnetic condenser lens. Specimen is there. You have the objective lens. This is all electromagnetic. Even though they have written as lens, they are all electromagnets. Fine. You have got an intermediate electron magnet. You have a projector lens, and then you are getting a fluorescent screen. Fine. So you can see how the light is traveling. I mean, how the not light. Please excuse. A beam of electrons are traveling in the transmission electron microscope. So these electrons, which is produced by the transmission electron microscope, are focused on to the specimen, and as a result, we will explain in the next slide what is actually happening. Right? I'm not going to explain from the slide. No, this is the way by which this is an example for an image produced in transmission electron microscope. The image will appear like this. We can clearly visualize the molecular arrangement. So from this picture itself, we can see what are all the internal structures are present in the microbial cell. Clear? And now we are going to the second type of electron microscope, scanning electron microscope. Fine. In transmission electron microscope, the electron beam is transmitted from a hot tungsten filament, which is directed onto the specimen by means of a set of electromagnets. Clear? Whereas in scanning electron microscope, the electron beam is scanned across the specimen. So not just transmitted, it is scanned across the specimen surface. So what is happening here? First of all, similarly to there, you will have a hot tungsten filament and a beam of electrons are produced. This primary electron beam knocks electrons out of the specimen. So there the electron in transmission electron microscope, you have got this electron beam which is produced from the hot tungsten filament, which passes through a series of electromagnets and focused on the specimen, and image is produced. Whereas here, the electrons which is produced by a hot tungsten filament are called primary electrons. Now these primary electrons will produce or knocks electrons out of the specimen surface. Okay, the primary electrons will go and hit the specimen surface, so that electrons are produced from the specimen surface, which are called secondary electrons. Okay, now the secondary electrons which are produced from the specimen surface are transmitted to a collector. Now this from the collector, this beam of electrons, or the, so here what is happening? First of all, hot tungsten filament produces a beam of primary electron. This primary electron will go and hit the specimen surface and produce a beam of electrons called secondary electrons, which is produced from the specimen surface. Now these secondary electrons are transmitted to a collector. From the collector, it is directed to a detector which emits light when struck by electrons. Okay, so here light, so similar to this happens in your transmission electron microscope, but there is no secondary electrons in transmission electron microscope. You have got the electrons produced from the hot tungsten filament in transmission electron microscope, which is directed to the specimen surface. Whereas in scanning electron microscope, the primary electrons which is produced from the hot tungsten filament will hit on the specimen surface and produce the secondary electrons. So in transmission electron microscope, the electrons which is produced from the hot tungsten filament, that is the primary electrons, are responsible for producing light. Whereas here in scanning electron microscope, the secondary electrons which is produced from the specimen surface are responsible for producing light. Rest everything is almost the same. Clear? So here the secondary electrons are transmitted to a collector, directed to a detector which emits light when struck by the electrons. Clear? Now this emitted light is converted into an electric current. Fine. In scanning electron microscope, the emitted light is converted to an electric current which controls the brightness of image on CRT screen. So what is CRT screen? CRT screen is nothing but cathode ray tube screen. CRT screen means cathode ray tube screen. Clear? So this secondary electrons will be directed to a collector and detector, and the detector will emit light when struck by the secondary electrons. Now the emitted light is converted into an electric current which controls the brightness of image on CRT screen or cathode ray tube screen. So that means if the amount of secondary electrons, so it depends upon the specimen. If the amount of secondary electrons are high, you will be having more light produced and you will be having more electric current produced. So as a result, the brightness of the image will be higher. It's all depends upon the specimen, the composition of specimen or the thickness of specimen, or how well we make the specimen. That is most important. Fine. Now the intensity of image reflects the composition and topography of specimen. So we will get a proper how the specimen looks like. What is the composition of specimen? What are all the various biochemical layers? Its composition. Topography. Topography means surface structures of specimen. What are all the surface structures of specimen? Fine. Which in turn determines the contrast. Okay. So the intensity of image on the screen reflects the composition and topography of specimen surface, which in turn determines the contrast. So all these things are related. Fine. If the beam of secondary electrons are more, the amount of light produced will be more, the amount of electric current produced will be more. As a result, the brightness of the image will be higher on cathode ray tube, which means the intensity of screen will be higher. As a result, you are getting a higher contrast. And it all depends upon how well we make the specimen, what is the composition of specimen, topography of specimen, that is what are all the various biochemical layers present in the specimen, what are all the various surface structures present in the specimen. All those things will determine the contrast, final contrast of the specimen. And finally, final point, scanning electron microscopy is used to view surface structures and their three-dimensional appearance. For example, if we are observing a flagellated microorganism, we can very well observe the structure of flagella and its three-dimensional appearance in scanning electron microscope. So scanning electron microscope is basically used to view surface structures and their three-dimensional appearance. So now I hope you have understood the difference between transmission electron microscope and scanning electron microscope. In transmission electron microscope, electrons are just transmitted from hot tungsten filament, which is directed onto specimen, and the rest is similar. Whereas in scanning electron microscope, the primary electrons produced from the hot tungsten filament will produce a beam of electrons from the specimen surface called secondary electrons, which is responsible for the formation of the image. So since these secondary electrons are produced from the specimen surface, we will get a scan of the specimen. A scanned image of the specimen can be produced using scanning electron microscope. As a result, we can visualize a proper composition and topography of the specimen, as well as three-dimensional appearance and also the surface structures. All those things can be viewed using scanning electron microscope. This is the how. So you have got an electron gun here. Fine. Electron beam is produced. Clear. And it is passing through few magnetic lenses and the backscattered electron detector. You have the specimen C. So from the specimen till the specimen, you have got one view of electrons. From the specimen, some of the electrons are backscattered, whereas the electrons which is produced from the specimen will go to the stage where the image is produced. Clear? And see, this is the way how image appears in scanning electron microscope. You can very well visualize the 3D appearance of surface structures. We can see from this picture. Please explain topography. See, now you can see this picture. What do you mean by topography? How the specimen looks like? Topography. Topography means it's like what I can how I can explain. Topography means, now you can see from the picture. You can see, now you can visualize some round bold specimens with spikes. You can see here you can see round bold structures with spikes. You can see some oval structures with some pores. You can see some round structures. Yeah, it's like morphology, arrangement, everything. It will all come together. Like topography means how a specimen looks like. What are all the various structures we can see? The size of the structure, the shape of the structure, whether you have any surface structures, all those things will come together in topography. You can see from the picture, what is the topography of this specimen? You have some round bold specimens with spikes. You have got some triangle-like structures here. See, here you can see some triangle-like structures as well. Fine. You can see some oval structures here, a group of oval structures. You can see, and even various colors you can visualize. Fine. It's all depends upon the topography. What is the composition of various? That's what in the beginning I told you. When we tell about various biochemical layers, what are all the various biochemical layers? Whether you have got a thicker peptidoglycan layer, thinner peptidoglycan layer. Fine. Whether you have got some phospholipids on the cell, whether you have got some spikes on the cell surface, whether the organism is flagellated or not. All those things, all these details can be visualized using electron microscopy, and especially in scanning electron microscope, you can observe the 3D structure, three-dimensional structure, how it looks like. Clear? So that's all about electron microscope. That's all about the unit microscopy. I hope I have covered almost all points in your syllabus. please go through the syllabus also, compare it with whatever I taught. If you need any further explanation in anything, I am ready to do that. And I have not prepared any questions today, since I wanted to complete this unit today. So that's why I took a little more time. So the time is almost 7:15, 7:10, and now you can expect the questions on Saturday. I will poll the questions in our Telegram group. Please do those questions so that you will get more information. Fine. In each question, I am giving some explanations if it is needed. I am giving some explanations in the question section itself. Once you answer the poll, once you provide your answer in the poll, you will get the correct answer. What is the percentage of correct answer told by you people who all have made the correct answer, who all have made the wrong answer. Now, if you have any additional information about the question, also see the question itself or the poll itself is self-explanatory. That's why I am doing polling in our Telegram group. I hope you are doing those things. You are attending those polls regularly on Saturdays. Fine. So that's all about the topic microscopy. And if you have any doubts, anything you come across, please do let me know. Already I have given my email ID and my WhatsApp number. Even you can send WhatsApp message or even mail if you have any doubt in your topic. Okay, I am always available with Biosetup team to support you like all other members. Fine. So that's all about today's class. Over to the organizers. And if you have any doubts, please let me know. Thank you. Once again, wish you all a very happy Dasara.
Okay, sir. And students, if you have any doubts regarding today's session, you can just ask in the comment section. Just write your questions in the comment box.
Shivam, are you still in the temple?
No, sir. No, sir. I returned. I am in home now, sir.
Okay, fine. No, just I want to know, that's why I asked.
Okay, sir. So students, if you have any doubts regarding today's session, you can just ask questions right now in the comment section.
So as Shivam has,
if you have understood today's topic, you can.
Yeah. As Shivam has explained. As Shivam has explained. So we will be sharing the slides with the registered participants, right? That is what we are planning to do now.
Yes, sir. Yes, sir.
Yeah, and even the presentation is available in YouTube and even our Telegram link everywhere it is present. So that's fine.
Yes, sir. We have a proper plan to share all those things to our registered candidates. Because sharing PPTs in now itself in the group, this will be not continuous. If once we start sharing all those PPTs in a regular way to all those registered candidates, then it will be much better, sir.
Yeah, exactly. No, that's up to you. I just asked what is our that's fine.
So if you have understood today's topic, you can just comment yes in the comment section. Just let us know. Okay, thank you for responding. Very enthusiastic aspirants. So I hope you all are going to register